So thank you very much for the nice introduction. I hope you can hear me okay. My presentation here today is about how to maximize viability, productivity of a CHO cell culture. And before I begin, I want to quickly explain what is all these words you see on the screen. So Ajinomoto and Genexine, two different companies, and the joint venture of them, Ajinomoto Genexine, is where I'm working now. Our main product is called Cellist, is the brand name. Cellist is short for Cell Specialist, and it's a cell culture media. OCS Lab is short for Ozone Customer Service Lab, and that's a lab and center and a service center located in Ozone. I will quickly show you the explanation a bit about OCS. So Ozone Customer Service Center is located here in the Ozone Bio Valley center of Korea. You probably heard about it. There are more than 200 different biopharma companies here. It's quite a large bio cluster here. And we are located in the SP Plaza. It's the Science Business Plaza. So let me begin with a quick overview of what I will be talking about today. I will quickly give an introduction about what does it mean biologic biosimilars, what is the biologic drugs market currently, and what is a CHO cell line, what does it mean, CHO cell line. I would also talk about very quickly about the biological drug development and manufacturing, and I will show a workflow of how to evaluate and develop cell culture media. And I will also talk about how to deal with the different performance issue that you might deal with when you culture CHO cells. I will also talk about cell adaptation, one of the important things that are needed when you switch from one kind of cell culture media to another. And I will talk about screening and optimization of cell culture media. Finally, I will talk a bit about things which are quite high-tech these days. They're calling things which include terms like Omics, computer modeling, big data analysis, even AI. All of these techniques or platforms are being used these days to optimize both process and the cell culture media used for production of biologics. Okay, so let's begin with a quick overview of what does it mean biologic drugs. So perhaps you know, but the current biologic and the biotherapeutic market is quite huge, over 120 billion US dollars per year. Most of it, around more than $90 billion sales, are only from those 10 drugs. You probably heard about many of them are anti-cancer or other autoimmune disease drugs. Now, there are different types of biologics, and there are also something called biosimilars, and I will quickly talk about the difference between them. First of all, what does it mean, biologics? So compared to normal traditional drugs, so you must know things like acetaminophen or aspirin. Those are traditional drugs that are basically very small molecules, and they are synthetically made or made by other microorganisms. Those are kind of compared to very small vessels like a boat. And this kind of diagram shows the increase in complexity when you move from a small molecule drug to a larger protein and a larger biologic drug, which is, for example, antibody, IgG. So the complexity increase, and when you are dealing with antibodies and biological drugs, you cannot really copy or synthesize the same as you can do with a chemical like aspirin. You actually have to use cells, mammalian cells, CHO cells, or other types of cells, and those cells are creating the antibody, the protein. So for production and costs, you can see here how the timeline quite different. So 2 to 3 years to develop a small molecule drug, and compared to over 8 to 10 years to develop a biologic or biosimilar. The price, of course, is very different too. Now, the biosimilar market compared to biologic market is increasing from year to year quite rapidly, because for every biologic drug that is out there, you have dozens of biosimilars competing, just waiting to go out to the market the minute the patent is expired for the biologic. So many of the famous biologic drugs, many of them are anti-cancer drugs, are being expired. The patent is being expired these days and in the near future, and that leads to increase in the total biosimilar market, which currently is more than 10 billion, but projected to be by 2025 more than $30 billion, just the biosimilar. Just for example, quite a few drugs like Humira, Remicade, many of them, each one of them has dozens of biosimilars. There's also something called biobetters. Biobetters are not exactly a copy or imitation of the actual original biologic drug, but it's actually trying to be better. So it's a modified version. Now, we are talking about CHO cells, so I assume not all of you are working with CHO cells. So I will quickly give an overview of what does it mean. Chinese hamster ovary, CHO cells, they are basically epithelial cell lines that derived from the ovary of Chinese hamster. It was derived many years ago, more than 50 years ago, and it's become a continuous cell line. Went through different modification, genetic modification during the years with different lineage, different types, different subtypes of cells. One important thing to remember is that most CHO lines used today, of course, in biopharmaceutical manufacturing are suspension cell lines. So they are not adherent like these CHO cell lines, but they're in suspension and they're floating in the liquid in the cell culture media. And it's important to remember that most biopharmaceutical these days, most biological biosimilars are produced by CHO cells. Now, these different types that if you are in this field, you probably heard. There are K1 cell line, CHO S line, DG44. Those are the three kind of main subtypes that are used. And they are different in different way in the selection system. They have different differences between them, but most of them can be used to produce any kind of antibody you would like to produce. A quick overview of the biological drug development and manufacturing process. Of course, I'm not going to go into all the details, but just to explain the first initial seed train, which means taking the first initial stock of the CHO line, of the CHO cells, putting them in flask, increasing size of flask, different bioreactors, which with increasing size to increase the total mass of cells until reaching the final bioproduction bioreactor, which could be thousands of liters big. After that, there are lots of steps, complicated and expensive steps for purification, for extractions, for removing viruses. All these steps until the packaging to get the final biopharmaceutical that goes to the patients, to the hospitals. Now, my company, as I mentioned, Ajinomoto Genexine, develops cell culture media. Cell culture media takes part at the initial stage of cell growth. This is probably the most important stage where cells are growing rapidly. They produce the antibody, the protein or antibody target. A lot of quality attributes, so the quality of the final product is being affected by this process and by the cell culture media used. This small illustration here shows that also during cell banking, we also use cell culture media at different steps, making the WCB, the working cell bank or master cell bank that being used later for production in large scale. So the basic workflow when you evaluate, when you develop cell culture media is like this. Of course, it changes, depends on the manufacturer and depends on the stages and depends on the target proteins you deal with. But basically, it starts with adaptation, which means the cell line needs to be adapted to some kind of medium. After adaptation, there should be some batch culture. Usually, typically take between 1 to 3 weeks. The batch culture is usually for testing the cell culture media, or you can test different types of media from different manufacturers. There's also following up a fed batch test. So the fed batch test includes feeding. Feedings are supplementary media which is concentrated and added at low percentage to the bioreactor or the flasks, and then the reason for that is to increase the nutrients level, vitamins and amino acids and other nutrients that are needed for the cells when they are already the exponential growth phase and start producing the protein of interest. For the next stages, when you want to adapt the cell culture media and when you want to optimize it to your cell line, there's basic optimization and there's also more advanced optimization. Basic optimization includes finding the basically optimized feeding manner, the feeding volume used, and the timing, different process parameters that I will talk about later. And then advanced optimization looks at other different components that can affect the total performance and also affect the final protein quality, like CQAs, critical quality attributes of the final product. So I will talk about more advanced optimization methods later. Okay, so next I want to mention just some pointer or some important things to remember whenever you are growing CHO cells, mammalian cells. Those things are actually apply for any kind of mammalian cell culture, but some of the points here are specifically for CHO cells. How to ensure the optimal performance of the culture and the productivity, which means production of the antibody or protein. So recovery passages, those are quite important. So it's important to remember when you take a vial, a car vial from the liquid nitrogen or other deep freezer, when we want to start the cell line, it's important to first do a few passages, subcultures of the cells, before we start the actual main culture. That's because after the deep freeze, the cells take some time to get used to not being frozen. So it's important to do at least 2 or 3 or 4 even recovery passages before the main culture. Adaptation, sequential adaptation is often required when you are transferring cells from one media to another, and I will talk about it later more. The inoculation density is typically between 0.3, 0.5 million cells per milliliter, but that really depends on the style of process you are employing. But generally, when you want to adapt cells to new media or test different run tests on the cell line, you need to start an inoculation about these values, because you want to have the cells before they are in the exponential growth phase. And glucose is important because that's the energy source for the cells, the sugar, and it's important to maintain higher levels of glucose. Again, it depends on the cell line. Every cell line has glucose consumption profile, which is specific for that line. Growth factors is an optional, and some cell lines require growth factors, some don't. Insulin or insulin-like growth factor 1, other growth factors are sometimes required. So basically, if your cell culture seems to not go well and the viability is low, it could be one of those reasons. It could be the lack of growth factor. Glutamine is required, but not always. Depends on the selection system used. So you need to actually check the cell line and see if glutamine is required for that cell line. Other supplement like anti-shear, anti-clumping agent. Anti-shears, for example, poloxamer, which is often added to cell culture media, that protects the cells from shear forces, from stress. Anti-clumping agents prevent the aggregation of cells. And of course, if you add too much of those, you can get very low viability or cell death. So that could be another reason for a cell culture that is not performing well. Next one is the optimal temperature and pH. Of course, we always think 37 is the optimal and pH 7 should be okay. And that's usually the case, but the optimal for every cell line depends on the cell line. And during process, you have to optimize those parameters too. Osmolality is also important. So the ionic, the ionic pressure is important to maintain around this range. So again, if you have a poorly performing culture, you should also check osmolality, because that could be a reason for cell death. Also oxygen transfer, which includes the agitation and shaking conditions, that also affect the culture health. And of course, finally, keeping aseptic, clean and sterile cell handling to avoid any contamination, because contamination by bacteria or mycoplasma also can affect the cell growth. Now, I was talking before about media switch. What does it mean, media switch? Why do we need to switch media? So often, any cell line is growing in some original media. That cell line is used to this media. It's not necessarily the optimal. It's not necessarily the media that gives the best results, the best productivity. Other media could be better for that cell line, and then it's worthwhile to switch to that media. These are the consideration to switch to another media. Cost. The cost is very varying between different companies, and supply is also varying depending on the country you are, depending on shipments, especially now in the Corona world. Some supply chains are cut, and you have to take this into consideration. The consideration for media switching. So as I mentioned, every cell line has original medium, which it was developed and expanded in. And usually there's no problem to switch from one cell culture media to another, especially if it's the same or similar composition. Now, most CHO lines now are used or adapted to be in a non-serum containing medium. So most media used is chemically defined with no animal proteins, no serum. So they usually no much problem switching from one chemically defined media to another. The performance and productivity is very depends on the medium composition. So cell growth, the titer of the IgG amount, and also the protein quality parameters also affected by the medium composition. Now, there are two common techniques that are used to adapt cells from the original to a new media, direct and gradual adaptation. So I will quickly show you the kind of a guideline and the example of those two types of adaptation. But basically, direct means a direct transfer from the original media to the target media. That's always usually works if the two types of cell culture media are similar properties. If you do that and you find out that your cells are dying or are quite a low variable cell density, so you need to deploy a more gentle transfer way, which is called gradual adaptation or sequential adaptation, which means gradually you increase the amount or the ratio of the target media during the adaptation passages. So there are many types of way to do it, but this general layout is like this. When there is a ratio between the current, the original, and the new medium you want to switch to, you start with the original medium, 100 to 0, and 75%, 25% increase 25% of the target medium, then 50%, 75%, and 100% for every passage. Seeding density should be usually around this range, 0.3 to 0.5 million cells and per milliliter. And before continuing to the next stage, or before increasing the amount of your target media, or the concentration of target media, you should make sure that you have enough cell density. So it should be at least 1, 2, preferably more, a few millions of cells per milliliter, and the viability should be over 90%. Because if it's less than 90%, it means a lot of the cells died because of that target media, and you should take care of that and see what the problem is. And each passage takes about 3 to 5 days. Again, it's very cell clone specific. Some cells go faster, some slower. In general, there's no need to do the whole process of long sequential adaptation, because usually, in many cases, 2 adaptation steps are enough. You can do a 50-50% of original and target media, and after 1 passage, transfer to the final media. But if this doesn't work, you should employ more sequential most stages. Okay, so just show you a quick example. I mentioned CellList, right? CellList is the cell culture media which we develop here and sell. And in this example, we took a cell line that has some original media, is adapted to some original medium, and we directly put it in our medium, and you can see there's initial growth which is much lower than the original, which is red. That's almost always the case if you just transfer directly to a new media. But the moment you do one passage of 50-50% to adapt the cell to the new media, then already you can see growth which is very similar to the original growth curve of that cell line. If it's not enough, you can always employ more stages, as I mentioned. After you adapt the cells to a new media, maybe because you wanted to switch from one company to another cell culture media company, or you wanted to optimize the media. So after adapting the cells to that media, you want to run the tests and also optimize feed manner, optimize different process parameters. This list is basically kind of a stages, but it really depends on your process, and it really depends on your target. And usually it's just the stage goes like this usually. So the first is a basal media screening, so growth media screening in a batch process, followed by a feed media, and a feed media, basal and feed media combination. And the next stage is to actually optimize the actual feeding manner and volume. So how much concentration of feed and which days. Usually it's added every 2 days from day 3 or day 4. And the next stage is to start optimize different parameters, more physical parameters, process parameters like temperature, pH, agitation speed, and aeration. And that's being done in a more larger bioreactors, or using the microbioreactor like Amber. Finally, there are more complicated optimization process that includes looking at different components and different amino acids like cysteine and tyrosine, for example. Tyrosine has a very low solubility, so it requires different type of treatment, and cysteine can oxidize quickly, so it's not stable. So different kind of feeds, including cysteine, tyrosine, or sometimes they are combined with a main feed, are being used and need to optimize the concentration. Other supplements, for example, anti-clumping agents like DSS, dextran sulfate salt, shear protectant like poloxamer or other growth factors or growth factor analogs being used, and needs to be optimized. If too much of them, they can cause a toxicity effect and reduce the cell growth. So that needs to be optimized too. The amino acid composition can be optimized, and also all those components and other media components can be optimized to get the best quality, protein quality attributes. So especially when you develop biosimilars, you need to match your protein, your final antibody or protein, to the original biologic drug. There are glycan profile and charge variance attributes that require to be within the correct range. So compositions of amino acids and other media components will also affect the glycosylation and charge variance. So all of this optimization are done either in Flux or microbioreactors like the Amber. Now, talking about the Amber, so the Amber is kind of a standard way to develop a process, and it's very useful because it allows to check and to test and to optimize many parameters, including the composition of basal and feed media, different supplements, as I mentioned. You can optimize the feeding volume and the timing, the temperature, the dissolved oxygen. Seeding concentration also needs to be optimized, and as I mentioned, all the composition of the media. And the Amber is quite comparable to larger bioreactors. Of course, it's very small. It's bioreactors which are only maximum 15 milliliter. in this amber system, but it has many characteristics that are similar to large 2 and 3 bioreactors. Okay, quickly showing you kind of a image of different parameters that are being tracked during a culture, either using an amber bioreactor or using a flask culture. Those kind of parameters are usually monitored during the culture. For example, this example looks at a couple of basal media, growth media, and feed media combinations, and looking at a few different concentrations of 2, 4, and 6%. Now, it's not so important. I know the text is not so clear, but it's not so important. I just want to show in this slide how those kind of curves and graphs looks like and why they are important. So, of course, VCD is a viable cell density, and we want something that goes high enough and not showing such a low growth, and viability should also be maintained as much as possible. Titer, of course, is one of the main considerations, which means the IgG concentration, and of course it increases during the culture. Lactate, ammonia, and glucose, those are important to monitor because glucose should always stay above 0, shouldn't be depleted, because the cells really need it for the energy source. But glucose is being metabolized to produce lactate. Lactate as well as ammonia are kind of toxic byproducts. So lactate level and ammonia level should be reduced to the minimum. And it really depends on the cell line, but it should be like in this example, you can see when one cell line is producing a bit too much lactate, so we will not choose this combination of basal and feed. Other ones are kind of within standard, less than 3 grams per milliliter, or 2 milligram per liter. Ammonia level also shouldn't be too high. So of course it's produced during the culture, but it's better when it's not too much of it. Again, as I mentioned before, the feeding manner and all the other culture and process conditions are really specific for the cell line, and not only the cell line, the cell clone itself. There could be 2 different clones of CHO-K1, for example, which behave very, very differently because they were completely from different sources, and had different kind of modification during the years. The protein products also is something that will affect the parameters, that the best parameters for the culture. Next, I want to just quickly mention about an issue that sometimes happen. So some of our customers sometimes report about aggregation. So cell aggregation can happen and should be avoided. It can happen. Mostly the reason is because of cell lysis. So cell lysis releases the DNA, and DNA is a bit more sticky, and it causes aggregation and clumping of the cells, like in this picture. If there's a bit of aggregation, it doesn't matter. But medium A looks fine. Medium B here looks quite clumpy, so it should be taken care, and anti-clumping agents could be used, like descan sulfate, and the adjustment like the speed and other culture condition will be used to increase the variability. There's some noise on the background, but yeah, okay. Very quickly, because we don't have too much time, I will mention about protein quality attributes. So generally, glycan profiles means the glycosylation on the antibody. As you can see here, there are different types of glycans that are being attached, different type of sugars, and there are many media components that will directly affect the glycosylation. Why is it important? Glycosylation will affect the actual efficiency of the drug, the folding of the protein, the cytotoxicity, many attributes of the final protein drug, for the protein, will be affected by the sugar chains. So sometimes if biosimilar developers need to modify glycosylation profiles, they go to the media and look at different media components and adjust the composition in order to match the right glycan profile they need. And that's, if you're interested, you can see the link here, but there's quite a lot of information, literature you can find about different components and which kind of glycan profile they are affecting. So I won't go into these details now. I want to talk a bit about the approaches for media composition and optimization. So one approach is to use DOE, design of experiment. And there's a few ways to go about it. So if we consider designing a composition for the cell culture media from scratch, it means that we need to test more than 50 media components, because that's the amount of components in typical cell culture media. It includes, as I mentioned, amino acids, vitamins, salts, sugars, trace metals too. It's almost impossible to test all the combination and all the concentration because it would take thousands of combinations of all of them. So usually optimization focuses on a few of them. In the second approach, we basically select the most important candidates. How we find those candidates to do a DO test on them, that's something I will talk about later, and that involves more looking at metabolic pathways and omics approach to finding the media components. But basically, the idea is to choose components that could be bottlenecks in the process or limiting factors, which means the concentration will more directly affect some metabolic pathway that relates to the productivity of the process or the cell growth. And as I mentioned, using a computer modeling, a big data analysis approach, and multi-omics approaches, you can identify those. You can find those critical components and bottlenecks. As I mentioned, it's quite a continuous process to optimize the best cell culture media, and it's very specific for the cell clone, the cell line, and the protein product. There's no one solution that fits all. Now, I will show you a quick example of media enhancement experiment we recently did here, but I'm not actually showing you all the concentration. It's a bit confidential, but basically it's a standard DOE, design of experiment approach, looking at factorial design, which includes 4 factors with 2 levels. Factors mean, for example, 4 different amino acids. Concentration was varied. 2 level means at a high dose, a low dose, represented by plus and minus. All this combination makes a full factorial design that goes not only to see the effect of each amino acid by itself, but also correlation between amino acids. So basically, we use an instrument which is a robotic dispenser, powder dispenser. We can use it to accurately weigh the media, the powder media, and create unique combinations, which is then using the Amber, the microbioreactor system, to test on a cell culture. And then follow through analysis of metabolites as well as all the amino acid consumption rates. And that's basically the DOE. Later on, we can actually do a multi-omics analysis, which I will show later. So for the DOE, we use, for example, the JMP statistical analysis software. It's quite common way. Again, I'm not going into details here, but using this analysis, we can see the effect of 2 amino acids and also cross-correlation effect, how the synergetic effects between them. If one is increased, it relates to increase in titer, for example. But if the other one is increased or decreased, it will affect the effect of the first amino acid. So there's always correlations. And you can make this kind of prediction plots that gives you a prediction of which amino acid to increase and what kind of titer you can be expecting according to this composition. So again, I'm just touching it quickly. I want to go back to the to the discussion about the media composition, just to kind of how do you say go through the main issues and that consideration that we need to look at when we modify media components. So, as I mentioned before, this condition will affect the cell culture, and we need to consider them. We also need to consider things like solubility and stability. So tyrosine is usually very low solubility, and often dipeptides are used, like glycine tyrosine, because they are soluble and they can be uptaken by the cells to use as a tyrosine source. Stability, like cysteine and other components, which be affected over time also. And the cost is a really important thing. So when you design your media, you need to think in large scale. Amino acids are very cheap, for example. Vitamins are expensive, and other components could be more expensive. It's important to remember that the media components, they affect the overall process. Some media components may have an effect downstream on the purification steps. So you need to consider that if you have some problem downstream, you should look upstream at the media and think if some component could have caused that. And of course, safety and handling, shipment, availability of the raw materials will also affect the decision for your media composition. And I was talking about all this idea of CQAs, critical quality attributes or KPIs of a process. So kind of just general idea of what it means. So we are talking about CHO culture process, and the key performance indicator, what usually are called as KPIs, culture performance is considered a key performance indicator, which include the density and viability of cells and productivity, of course, the protein titer or concentration. They are also sometimes referred to as CQAs, as a quality attribute, although they don't deal with the actual quality of the product. So actual CQAs or critical quality attributes are the glycosylation profile or charge variant, the immunogenicity, toxicity, or other attributes of the final protein drug. And just to mention a few trade-offs when they are dealing in bioprocess. So of course, we assume that culture growth and density is always good to get as much VCD as we can, as much cell density, but we have to remember this: if all the energy of the cell goes to proliferation and cell division, then less of the energy goes for the actual production of the protein target. So there usually is some kind of a shift and a trade-off between how much cells we get and how much product protein we get. There's also a trade-off between the culture length and the productivity. So some processes in the industry are actually not 2 weeks, but shorter. Cells are inoculated in high density sometimes in order to reduce the total culture, and then more cycles can be done per year to increase the productivity. So that's other trade-offs. And of course, cost versus performance. So you have to consider, like I mentioned, the cost of all the raw materials, components, and different equipment in the upstream and downstream. And finally, I just want to show a few slides about what I mentioned before about omics. So omics and general computer modeling approach for media and process development is something that existed for a few years, but recently become a bit more important. And there's also artificial intelligence algorithm being used for this kind of analysis. So, for example, we in my lab, we are collaborating also with groups who are kind of expert in multi-omics analysis. And the difference is that traditional experiments, for example, DOE approach, are mostly statistical approaches, looking at all the data derived from a wet experiment and trying to find the best combination and correlation to get the best performance. But the new concept of omics is mainly integrating statistical analysis together with mechanistic approach. And I will explain about that quickly. Generally, the benefits of omics, so everything that from metabolomics to transcriptomics, big data analysis, all the benefits of it is to better represent the cell, to get a model of the cell, understanding all the metabolic pathway, reduce the degrees of freedom, basically reduce the number of unknown of the process, because the cell is kind of like a black box. So we want to model, simulate what's happening inside it. To identify the rate limiting and the steps and the bottlenecks, and looking at basically the effect on productivity as well as effect on downstream protein quality characteristics. And the process, again, just in general, is getting all the data from cell culture and then process the data to find the residual concentration, to find the consumption rates of all the components, including amino acids and other metabolites. From there, there's a whole process of elemental balancing, which means looking at carbon, nitrogen, oxygen levels, and basically considering also the amount that goes inside the cells and the amount that goes outside of the cells in the form of metabolites and also in the form of the actual protein products. And when you do all this balancing, by getting the data from the metabolites, when you do the balancing, you can actually verify your data. So it's kind of a data integrity and confirmation step, or data quality confirmation step. After you get all these consumption rates and element balancing, you can continue into the analysis, which include the data-driven model using statistics, multivariate analysis, DOE, and the different statistical techniques like partial least squares and PCA. And this find correlation between consumption rates of different metabolites, but it doesn't consider the actual metabolic pathway. What's most important is the mechanistic model. The mechanistic model looks basically like a system biology approach to imitate or to simulate all the metabolic pathways happening inside the cells. And omics profiling usually considers more than 7,000 biochemical reactions in hundreds of metabolic pathways that are being modeled here. And then all the data is being gathered and designed and engineered in new culture media with improved process parameters. So this kind of cycle of optimization. And I will show you quickly with quick case studies how it was used. Before that, I'm just showing you just kind of what kind of data you can get out of this kind of omics modeling. You can get all the, after calculating all the consumption rates of different metabolites, amino acids, you can actually get kind of data tables such as this, including thousands of individual biochemical reaction in pathways, and looking at which amino acids participated and where was a bit of a bottleneck, or which pathways were a bit like affecting the flow. And that's called the MFA, a metabolic flux analysis. And you can visualize the whole network of metabolic pathways to look at to identify where was actually bottlenecks in the process. So I mentioned quickly one example, okay, study that we did, looking at the omics approach to enhanced media optimization, enhance the amino acids. Here we compared two cell lines, cell line A and B, with two different media. So when we compare two cell lines with two different cell culture media, we are looking at different correlations and using the PLS approach to look at correlation between all the amino acids. And then that's the statistical approach, basically. And then we combine this with the mechanistic approach, which is a MFA, metabolic flux analysis. And we compare the central metabolism in this case, and we look at the magnitude of flux, which was a bit different. Again, not going into all the details, just want to show you quickly, and I will explain a bit more here. In this case study, for example, we used also omics analysis, but here we looked at a different type of process. We compare a process that happens in a flask to one that happens in a reactor. So if you can see, when the cell culture was going inside the flask, which is black line, it shows better performance than when it was in a bioreactor in a red line. So the black line shows higher growth, but actually not higher IgG production. But bioreactor shows quite increased lactate formation, as you see. So we tried to understand why was that, because the same media was used, but just different scale, flask or bioreactor. So comparing all the consumption rates of all amino acids, feeding into the mechanistic model to find metabolic flask analysis. And in this case, we found that the glycolysis cycle, or more the TCA cycle, was reduced in its effectiveness compared to the glycolysis chain. So for example, here, the process in the reactor shows a bit narrow flux in the TCA cycle compared to what happened in the flask. And glycolysis was a bit more flask here. So there was a kind of a bottleneck in this area, and the conclusion was then that not enough oxygens went inside here, and increasing the aeration of the bioreactor actually improved the conditions. So I kind of finished the main part of the presentation. We don't have too much time, so I just want to mention about us, the OCS, our center here, what we offer to the customers. Everything that I showed before, this stuff we did here. So we are basically providing support and consultation for any kind of cell culture media requirements. We usually get cell lines from customers and provide cell adaptation in here. And we have Amber and other equipment that is used for cell culture and fed batch optimization. Also, we have a protein analysis equipment that allows to basically affect the glycan profile and charge variant and other attributes. And we also starting to give more scale-up support, and in the future get more larger scale bioreactors here. And the overflow is, like I mentioned before, making some kind of powder media formulation and using either flask or Amber bioreactor for cell culture. All the analysis is done with this equipment. Spent media analysis done with UPLC also, and amino acid analysis, and metabolite analysis, and protein quality analysis. So we have quite a high throughput equipment here for analysis of the protein too. And then we employ also the multi-omics approach to optimize the media. And I think I'm over time here, but finally I just show you a quick video here of our, oops. This is the robotic powder dispense system that you can see in this picture, all the amino acids, not all of them, but in this case, we optimized 10 amino acids. There are 10 vials of amino acids, and we can make any number of combinations of ratio and compositions of those amino acids. And the advantage in this robot is that it basically prevents any source of human error in mixing and in weighing, and the sensitivity and accuracy is very, very, very high. So here it's put the vials here, and then it goes inside, and it's basically start dispensing the media down to accuracy of milligrams. And next one, I was talking about the Amber. So the Amber, the microbioreactor is basically looks like this. There are 24 small bioreactors here, and there's also a 48 version one. This is completely automatic system that feeds, for example, now it feeds the vessel, and it also measured the dissolved oxygen, pH, temperature, and maintains them. And everything is done also while counting the cell viability with this counter. And that's it. this is my last slide, and we actually changed the lab recently. We expanded, so this is a bit old picture. We are here ready to help anyone who wants our services to test the cell line on different media formulation, and we're actually also looking for new talent. So if you have any experience in media development in CHO cell lines, so please contact me. So thank you very much.